The overall goal of the following procedure is to evaluate the invasive ability of cancer cells using a three-dimensional invasion assay. This is accomplished by first culturing cancer cells in a basement membrane matrix. The cellular invasion of the matrix is then observed for 5 or more days via light microscopy. In the final step, the cells are labeled with fluorescent antibodies for localization of the proteins of interest. Ultimately, the rate of cancer cell invasion and the resulting changes in protein expression can be analyzed by immunofluorescent microscopy.
The main advantage of this technique over existing techniques such as the transwell chamber invasion assay is that the 3D matrigel invasion assay better mimics the in vivo environment that cancer cells grow in. The implication of this technique is towards cancer therapy, where new proteins can be identified and studied which are involved in cancer invasion and metastasis.
Before beginning the culture procedure, place the basement membrane matrix, a P200 pipette, and pipette tips on ice overnight at 4 degrees Celsius. The next day, use the tip of the ice-cold 200 microliter pipette to spread 50 microliters of the matrix in a spiral pattern over the bottom of a confocal No. 1 glass bottom dish. Then place the dish in a cell culture incubator at 37 degrees Celsius with 5% carbon dioxide for at least 30 minutes. While the matrix is undergoing solidification, trypsinize a 70 to 80% confluent 100 mm plate of cells. Once the cells have begun to detach, inactivate the trypsin with 10 milliliters of medium